Welcome, dear students. Today we're going to start to continue the chapter 5, DNA and genetics. And today, inshallah, we're going to start to study the structure of the DNA and how does it affect on our body and the whole entire living organism. Bacteriophages. In the last episode, we got some information and we discussed the experiments that give us evidence or clear-cut evidence that the DNA is the genetic material. And also we discussed some information about bacteriophages, and we said what does it mean, bacteriophage? It's a type or kind of virus that has ability to infect another type of bacterial cell. So it's a type of virus. Particle consists of what? A molecule of DNA that coated or covered, enveloped by a protein coat, as we mentioned it in the last piece of information in the last episode. And also, we said this type of viruses, it has ability to attach to the bacterial cell. And in our experiment, our phage or the bacteriophage, which consists of DNA molecule, and also there is an extension from this DNA or from this coat, which is called a tail. Okay, something like a tail. Just the bacteriophage uses this tail for attachment by bacterial cell, and also it observed that after 30 minutes of the attachment or after the infection of bacteriophages by the after infection or after attachment phage or bacteriophage to the bacterial cell, what happened? The bacterial cell, they are burst. They are absolutely explode. The bacterial cell burst and comes out of more than 100 new complete bacteriophage. After that, it was said that, or it's very obvious that some material or a group of materials of DNA passed from the virus to what? To the bacterial cell. Okay, so to inside the bacterial cell over, and they are already confused or merged with the DNA or the nucleus of the genetic material of the bacterial cell. So the bacterial cell, and that causes what? The formation of new viruses. What happened for formation of this new virus? The material which enters to the inside bacterial cell, they must contain the virus genes. So the scientist here, he concluded something, that the transferring of this material from virus, which is bacteriophage, to the material of the nucleus of the bacterial cell, it must contain the virus genes. It must contain what? The features or characteristics of the virus. And this material, it knows that DNA, deoxyribonucleic acid. So it contains phosphorus in this structure, whereas proteins doesn't contain phosphorus in its structure. There is no phosphate group in the protein structure. So the protein also contains a sulfur in its structure, but DNA it doesn't. So we have here a great difference between the structure of DNA and the protein. And it was very easy to prove that the DNA is the genetic material in all living organisms. By what? By isolating the DNA, okay, of the bacteriophage or of the bacterial cell. Also, Hershey and Chas, they made one of the greatest experiments. They use what? They use this fact okay, or this obvious experiment in carrying out an important experiment, which is what they labeled fake protein. Labeled here, it's I mean they give it a kind of marks or signs, okay, the bacteriophage proteins, by a kind of radioactive sulfur. Do you know that we have some elements, they have ability to be radioactive, or to emit what? To emit a kind of radiation, okay? We call them radioactive, and they are called the isotopes of the element, okay? Well, the radioactive sulfur, we have just the normal sulfur, and we have another isotope for the sulfur, which is radioactive sulfur. So Hirsch here, he already labeled or marked or signed the protein of phage by radioactive sulfur, and the phage itself, DNA, by radioactive phosphorus. So we have what? Two radioactive elements, radioactive sulfur and phosphorus. Well, they also allow the bacteriophage to attack bacteria, okay, the bacterial cell, and they determine the radioactive of each of two both elements, the sulfur and the phosphorus. The phosphorus and radioactive sulfur, they already are inside and outside the bacterial cell, and after that they are going to follow the passage of each radioactive element, phosphorus and sulfur, which one of them will remain inside, or which one of them merge with the genetic material or the nucleus or DNA, and which one of them it remains outside or within cytoplasm. This is the experiment, or the infection and attachment of the bacteriophage by the to the bacterial cell. Okay, on the left side here, this is the structure of the bacteriophage. It consists of, it's a kind of viruses particle, contains this is spiral. This head, which is hexagonal form, it contains the genetic material of the virus, which is DNA, and this coat, okay, is created or made up of proteins, okay, and this still, it has an important function for what? For the attachment by the bacterial cell. And on the right side of the screen, we have here about 6 steps. Okay, the first one of 6 steps of the infection or the attachment of bacteriophage to the bacterial cell. The first step, the attachment, okay, by using the tail. Okay, the second step, injection. It's genetic material to the bacterial cell. Okay, after that, after 15 minutes, what happened? The head already disappeared, and there is no any function for the head right now, but the genetic material, it starts to confuse or to merge with the genetic material of the bacterial cell. And after time, after 28 minutes, the each genetic material, okay, each new genetic material surrounded by protein coat. Okay, and finally, after 30 minutes of the attachment, the bacterial cell are what, as we said, explode, or they are burst, and about more than 100 new bacteriophage, completely bacteriophage, they are released, and the bacterial cell itself, it's already disappeared. Okay, well, the results of this experiment showed that all viral DNA, okay, or viral DNA almost enter to inside the bacterial cell. They are what? The viral protein, they didn't. So again, most of the viral DNA already they entered inside, while the viral protein, okay, I mean the proteins of the virus, of the protein, viral protein here, it's mean what? A protein of the bacteriophage, viral protein. They didn't enter to inside bacteria, except less than what? 3% of these protein. Just try to squeeze your mind to what? To make the coat of the DNA, to make the protein coat. Which means that DNA entered to the bacterial cell, and it push or it force it to build the new viruses. So here, after using the radioactive elements, sulfur and phosphorus, and also after the following steps of infection and attachment of bacteriophage to the bacterial cell, they concluded and they have a final cut evidence that the DNA is the genetic material in the all living organism. The conclusion, or this final answer, this evidence from that the bacterial transformation experiments and the experiments on the bacteriophage itself is that the genes, at least, okay, those of the pneumonia bacteria, this type of bacteria, and the bacteriophage are composed totally of deoxyribonucleic acid, or the DNA. But note that we also restricted that this conclusion, this final evidence to that the living organisms on which the experiments were carried out and followed some questions is what? We have a question here. Do you think that all genes composed of deoxyribonucleic acid, does all gene or any gene composed of DNA? We need to answer for this question. The answer actually to this question is no. Because of what? There are some viruses which they absolutely don't contain any kind of DNA in their structure, but they are contained what? RNA, ribonucleic acid. So we can conclude also, or we need to get some or carry some experiment to prove that the DNA is already, for the most of the living organism, is the DNA. But contain, as we said, maybe RNA, and also it was proven that from long time that the genetic material in these viruses is RNA. So because of this disturbance that we have different types of viruses, as we said, some viruses, they are composed mainly of DNA, okay, coated or uncoated, or as we said, it's naked viruses, and on the other side we have some viruses, they are composed of their genetic material composed of just RNA or ribonucleic acid, maybe coated or maybe not. So these viruses are exceptions, okay? The viruses, they aren't represent, they aren't express on the entire life, the entire structure of the living organism. So these viruses are what? Exception to that, this rule. So they are form a small part of the forms of the life. We need a clear evidence that the DNA is already carries the genetic material for all living organisms. And also, in view of the various studies that were carried out till now, till this moment, it is confirmed that DNA is the genetic material in nearly all the forms of the life. The quantity of DNA in cells. We're going also to make some information and to give you some experiments about that, the quantity or the total amount of DNA in the cells of a different living organism. Actually, there is an evidence that the DNA is the genetic material in eukaryotic. What does it mean, eukaryotic? Actually, we have two types of nuclei. Okay, this is a plural of nucleus. Okay, how many types we have? Two. We have some living organisms. Okay, they have their nucleus that's surrounded by nuclear membrane. Okay, if the living organism, it's a nuclei inside its body, or the body of the living organism, the nuclei surrounded by nuclear membrane, so we call it what? Eukaryotic. And on the other hand, most of the unicellular living organisms and some primitive forms of the life, the their nuclei, they do not surrounded by a nuclear membrane. Okay, so we call it what? Prokaryotic. Is it clear? Okay, so the evidence that the DNA is the genetic material in eukaryotic is what? Measuring the quantity, okay, the amount of DNA in different types or kinds of somatic cells. What does it mean, somatic cells? Somatic cells, any cells, okay, any cell inside your body except the cell of the reproductive organs, okay? I mean the cell that form your tissue and your body, except what? Except one type of the cell, which is reproductive organ, okay? Somatic cell of a certain living organism. And after that, such as what? The chicken, okay? It was found that they are equal, okay? But when the amount of proteins in the same cells was measured, it was what? It was unequal. So we have here what? A different formation. They aren't equivalent to each other. And by comparing DNA in the somatic cells or the body cells and the reproductive cell gametes, okay, as we started in the chapter 3 of the same living organism, yes, I need you to conclude the answer before turning the slide. Yes, the what? It was found that the quantity of DNA in the reproductive cell of the gametes, what? They have amount of DNA in the somatic cells. So scientists, they found that they got some confused or disturbance. And since the new individual results from mixing or combination of male gamete and female gamete, each gamete must contain half the genetic information that found in somatic cell or the genetic material. So studying the amount of DNA in each cell, it was a clear-cut evidence for us that the DNA, it's already the genetic materials, or it's already the substance, the material that carries the all genetic information for the entire life, for the whole living organism, okay, for the different various and different types of living organisms. And also, this amount will be duplicated in each generation, while that doesn't happen, this is not agree with the protein. This shape or this sequence, it doesn't happen to the protein, which proves that protein, it doesn't work as a genetic material. Protein not work as a genetic material. Proteins also and RNA are constantly being made and destroyed. They can be destroyed. But DNA, once made, is remarkably stable. For example, in every day in our body, we have a lot of mutations that may happen inside our body, okay? Well, for example, if any piece in your body, any muscle, it got injured, you hurt yourself, and a part of this protein, or like the muscles, they are already made of protein. If a part of your muscle is already injured, it's gonna be what? Repaired, okay? And replaced by a new one and a new type of protein. But in case of DNA, it once it made, it remains stable, okay, and doesn't change yet. And this type of stability, okay, it gives the scientists also evidences that the DNA is the genetic material that carries our genetic information. After that, we need to study the structure of DNA. Okay, the early 1950, there was strong evidence, there strong belief that DNA carries a cell's genetic information of any cell. And also, many people were trying to work out the structure of the DNA. So, after a long time, the scientists, after they got the final answer of the DNA is already, it carries our genetic information, they try to get inside this material, this magic material, which carries our genetic information, which is called DNA. So the scientists tried to what? To get the structure out of the DNA molecules and make a model for it. They wanted to do study it in details, okay, to decompose it. Any model of DNA structure must take into a consideration the following information, which came out from many experiments. I mean, after hundreds of experiments, the scientists, they put a list or criteria of how the model of DNA applied on these criteria or these steps. Okay, they are what? DNA is made up of nucleotides. Okay, and this nucleotide is made up of 3 parts. Again, DNA, it consists of its structural units. They are called nucleotides, and the nucleotide itself, it's made up of 3 parts, which are what? A 5-carbon sugar, or we call it pentose sugar. Pentose means 5, okay? Pentose sugar, or 5-carbon sugar. Sugar contains a 5 carbon. Deoxyribose in the case of the nucleotides in DNA. Deoxy, it's mean it doesn't contain oxygen. Deoxy, D, it's mean to take or to remove oxygen. Okay, so D, it's mean without oxygen. There is no oxygen in this kind of sugar. This ribose sugar, it doesn't contain what? Oxygen. So we call it deoxyribose sugar in the case of nucleotides in DNA, while the ribose sugar in RNA. Also, so we have here a 5-carbon sugar, phosphate group sugar connected with the covalent bond to the sugar's 5th carbon atom, okay? And one of 4 possible nitrogenous base. So we have here 3 parts. The deoxyribose sugar, okay, it doesn't contain oxygen, and phosphate group. And after that, we have nitrogenous base. And this nitrogenous base, in the DNA, we have 4 types inside the title of nitrogenous base. And this nitrogenous base containing what? Well, connected with the covalent bond to the sugar's 1st carbon atom. Okay, we're going to change the name of these sugars 1st and 2nd right now. The nitrogenous bases may be one of the single ring or the maybe double ring. The single ring, okay, we have 2, which are what? They are thymine and cytosine, okay? Or they may be double ring, such as what? Adenine and guanine. Again, we have 4 nitrogenous bases that the DNA includes: thymine, cytosine, adenine, guanine. T, C, A, G. And don't forget to write them with the capital letters. Yet, when the nucleotides are linked together, when they're connected together, okay, in a strand, in form of the ladder, as we're going to study it after time, to form the strand, the thread of DNA, the phosphate group attached to it, to the 5 carbon of what? Of the deoxyribose sugar of 1 nucleotide, okay? And this nucleotide becomes joined to the 3rd carbon of the sugar of an adjacent nucleotide. Adjacent, it means the closest one. We're going to show a picture here right now. This is the structure of DNA, and also we have the name of the scientists who discovered this type of DNA. Again, we have what? Each nucleotide consists of 3 main parts. They are what? Ribose sugar, or sugar of contains a 5 carbon, and phosphate group, and one of the 4 nitrogenous bases. They may be single ring or double ring, such as what? Thymine, cytosine, adenine, guanine, okay? and this is the form of connecting together. Okay, the thymine one, it connects with the adenine by double bond, while cytosine and guanine, or C and G, as we said, we can write the shortcut, but in capital letter. Don't forget it, with the what? With the triple bond. Okay. Well, after that, the strand in which the sugar alternate with the phosphate is called what? Sugar phosphate backbone. I mean, it's the backbone of what? The backbone of the strand. Okay, the main support of the strand. This is the mean here of that. The phosphate is called sugar phosphate backbone. This is the meaning of backbone. The backbone, okay, isn't symmetrical. I mean, it doesn't necessarily to be there's a symmetry in it. It has a definite orientation. So it has what? A definite orientation. Well, the prime 3 prime dash here is mean prime. Okay, we pronounce it a prime or dash. Don't say dash, but say prime. 3 prime hydroxyl group at one end, and a 3,5 prime phosphate group at the other end. Okay, so they are what? They are alternate to each other. Okay, I mean, we have two strands. Okay, over it looks like a ladder. Okay, the 3 prime in the opposite side, there's what? 5 prime, and also at the bottom, the 5 prime, the opposite one, the other stand, 3 prime. So there is no symmetrical, because there is what? There is a definite orientation to form the final structure of the DNA. The purine and pyrimidine bases, they stick out to one side of the sugar phosphate backbone. Okay, and also in each DNA molecule, the number of nucleotides containing adenine equals the number of containing thymine. Okay, and also I think that you are already concluded that the number of guanine, it absolutely equals the number of cytosine. So the total amount, the total number of nitrogenous bases, they are equivalent to each other. Okay, if we have 1 million of adenine nitrogenous base, on the other side they need 1 million of thymine nitrogenous base to be linked together, to form a double bond together. Okay, and in the same sequence, the guanine and cytosine. Okay, also the same number of the containing cytosine. Okay, that is adenine linked with a thymine by a double bond, while guanine and cytosine with the triple bond. Okay, the direct evidence that the DNA structure from studies made by Franklin. Okay, this scientist, one of the greatest scientists who proved that the DNA structure, or he gave us a final answer before the last one. Okay, this scientist used what? The X-rays diffraction. Actually, diffraction here, it's related to the light. Okay, to the rays. It's a nature of the light. Okay, like reflection, like refraction, but this type it's related to the kinds of rays that we cannot, they are non-visible. Okay, for our eyes, which is diffraction technology, to get the picture of highly purified DNA crystals. Okay. Our scientist, she wanted to prove that the DNA, it has a definite shape, or it has a specific shape. So she used the X-ray in the property of diffraction technology to get pure DNA crystals. What did she do? In this technology of X-ray, by passing the crystals or molecules which have regular structure, and result from it a scattering of X-rays. Again, the scattering of X-ray, it means that they aren't organized. There is no asymmetrical dots after exposing the molecules of DNA to the X-rays.